Hello there and welcome. My name is Brennan Brown and this is the genetics presentation for my Science 110 course at Sandhills Community College. In this lecture we are going to take a look at how our understanding of genetics came to be. We'll take a few minutes to talk about Gregor Mendel and how he first gave us the laws of genetics. We will talk about where genes are found in humans and how some of their traits are expressed, and we'll close with some of the current advances in genetics and biotechnology that are really impacting our world as we speak. We start with a gentleman by the name of Gregor Mendel. Mendel was this monk, religious type guy who lived in Austria in the 1800s. Gregor became very interested in trying to learn how organisms pass down their traits from one generation to the next. We don't really have to look very far to see that offspring often carry the same traits and characteristics as their parents, and that goes for almost all organisms, not just people, but animals and plants and um, fish and reptiles and everything in between. And so Gregor decided that he wanted to better understand the underpinnings and the inner workings of these characteristic traits that are passed down from generation to generation. And without even really considering himself a scientist, he undertook just a really spectacular set of experiments that, through his meticulous note keeping, he was able to really determine many of the kind of themes that we still rely on as our understanding of genetics today. What he did was, and perhaps it was a bit of serendipity that he chose to use pea plants, but he grew thousands of pea plants over many, many years. And while he was doing so, he selected them specifically for different traits. He looked at the color of the flowers on the plants. He looked at the color of the seeds the plants produced. He looked at the shape and size of the seed. Some were wrinkly and some were smooth. He looked at the size of the plant. So he decided to take these all of these individual plants with different traits and cross them and cross pollinate them and see how the offspring turned out and see how these different traits, these different characteristics mixed up and how they were expressed in the future generations. Uh, like I said, it was perhaps kind of lucky that he chose to use pea plants. They were a great candidate because of a few things. First of all, they grow really quickly, so you can perform a lot of experiments all at one time. They have and they had easily observable traits that came in just those two options. It worked out really nicely. He was able to gain some understanding because we only had two options in a lot of those traits that we just talked about. The flower color was either white or purple. There was no reds and greens and blues to worry about. Seed color was either green or yellow. Seed shape was either wrinkly or smooth. So those were very advantageous characteristics of the pea plant. And finally, the one of the last advantages that they had is that they sexually reproduce. So he was able to cross pollinate them and keep them separate, in which case they didn't pollinate themselves, which was important to the understanding of what he was trying to understand, the genetics of the plants. And here on this slide, we just have a representation of those different traits that he was concerned with. And you can see in the seed category, he looked at the form of the seed. So was it round or was it wrinkled? Was the coating on the seed yellow or was it green? And then of course we talked about how the flowers for pea plants can be either white or purple. Then he also had traits that were had a duality in the pods. You could have either a full thick pod or a wrinkled kind of sucked in pod. You could have different color pods. There was also differences in the placement of where the seeds were found on the plant and the size of the plant as well. Like I said, all of these traits, well, it was important for his research that these traits only came in these options. If it becomes a lot more complicated when you've got a lot more options, we'll look at the human genome later in the slides and talk about that for a little bit. Of course, something like human eye color, you can have brown or blue or green or hazel or black, or you can just that gets a little deeper than just two options. And so when he was studying this before any knowledge had been obtained of it, it was nice that he had picked an organism that only allowed for those two options, which really allowed him to understand the dominant and recessive factors of genetics. So with that said, let's take a look at some of the experiments that Mendel performed. In his, we say first set of experiments, what he did was for each set of traits, he had to have kind of a first parental generation. And so to study a particular trait, for instance, in the first set here, he chose flower color. Pea plants can have purple flowers or they can have white flowers. So he took a purple flowered plant and he crossed it with a white flowered plant. And he checked what color flowers do the offspring of those plants have. And what he found is all of the offspring of the purple-white cross had purple flowers. And then he took those purple flowers resulting from the cross and interbred them. He took the pollen from one of those first generations and put it on the flower of another generation, or of that same generation, and observed the characteristics of the second generation. And what he found was the white flower reappeared in that second generation. And furthermore, he did enough experiments to show that it always reappeared at a 3-to-1 ratio. So 75% of the second generation would have purple flowers and 25% of them would have white flowers. So for every three purple flowers in the second generation, you would get one white flower. And so Gregor Mendel, after observing the results of that first experiment, he proposed the law of segregation. He said traits in an organism, for instance flower color, have to be determined by two factors. To have this observation that I've made, that means that there are two kind of checkpoints in an organism that determine that trait. It's really interesting that he was able to come up with this. Remember, or keep in mind, he had no idea of what a gene was, of what DNA was, or what recessive and dominant was. He just said that there are two things that contribute to the determination of what flower color I got from my pea plants. Now he did say one has to be dominant and one has to be recessive. And for what that means is if both are present, we only see the dominant. So he said if both factors or if the dominant is there, then the dominant one shows through. If only shows the recessive factor, we only see the recessive factor shown if both factors there are recessive. And we do see that. That is one of our main underpinnings of current understanding of genetics, is that we have dominant genes and recessive genes, where dominant genes are expressed in the organisms much more often than the recessive genes. For instance, brown eyes versus blue eyes. Brown eyes is very, very dominant, and that's why we see more people with brown eyes, because most people have the brown eye gene, and if you have it, you show brown eyes. You can only have blue eye genes to have blue eyes because they are recessive. So he was able to make these descriptions again before having we didn't have any understanding of the cell or DNA, but he was able to say from his observations of the pea flowers' behavior that there were two factors that controlled what trait was present in an organism, and of those two factors, one was dominant and one was recessive. And that's what he called the law of segregation. So after determining that each trait had a set of two factors that determined what the expression of that trait was he wanted to see if traits themselves were linked together or were they independent of each other. And so to study that, he set up another experiment in which he decided to cross a pea plant that was dominant for both seed color and seed shape. So the dominant trait for seed color is yellow, and the dominant trait for seed shape is round. And so he took a completely dominant round yellow seeded pea plant and crossed it with a completely recessive seed plant for color and pea plant for seed shape and seed color. The recessive seed shape is wrinkled and the recessive seed color is green. And so to see those, you've got to have those two recessive factors, and both of them being recessive to get green wrinkly seeds. And like he did with the first set of experiments, he took these two parents and crossed them together to give us a seed generation or a plant generation that has one dominant factor and one recessive factor for both seed color and seed shape. So now, of course, because they do have the dominant factor, then they express the dominant traits. The whole generation had this configuration right here with one dominant factor and one recessive factor in both traits. But when we see the plants that it produced, because they all have that dominant trait, they only showed yellow round seeds. Now, of course, when we cross this generation, we get some that will have the recessive traits. And so we will see some of the green and wrinkly seeds appear again. But he wanted to know if some would appear yellow and wrinkly and some would appear green and smooth. And so we'll see what happens on the next slide. All right, so here we have the result of Mendel's second cross of that generation that were had the dominant and recessive factors for both seed color and seed shape. And this is what he found. He did lots and lots of experiments that mimicked or copied this experiment, and every time he got this ratio of seeds out of the second generation, and that was he got 9 seeds that were round and yellow. He got 3 seeds that were wrinkly and yellow. He got 3 seeds that were round and green, and he got 1 seed that was wrinkly and green. So he always saw the offspring of that second generation with these ratios. And that led him to understand that meant that different traits were controlled by different genes, and those genes are independent of each other. So sadly, the work of Mendel kind of went unnoticed for 50 or 60 years, and he never really got any of the recognition that probably should have came to him while he was alive anyways. However, once we expanded our microscope technology and we started to be able to see smaller and smaller with higher detail of the things that are going on in our cells, we started to evaluate the work that he did and apply it to learn how traits are passed on in living organisms. And that is done via something that we call chromosomes. So inside of the cells that make up you and every other organism here on planet Earth, we have a cell that it has a nucleus, and inside that nucleus is these long bundles of the DNA molecules, and they're grouped together in these bundles that we call chromosomes. And DNA tells cells how to make different proteins and different things it needs for energy and different structures within the body. It encodes all of the instructions on what cells to make and what types of things those cells should do. All human cells, in the absence of disorder, have 23 pairs of these chromosomes for a total of 46. We see chromosomal disorders resulting when some of these chromosomes are missing in cells or when extra copies are present. The most common of which is people who have Down syndrome have an extra copy of chromosome 21 in all of their cells. So chromosomes is where our genetic information is stored via DNA molecules. So during what we call regular mitosis, where a cell splits and new cells are made in your body, those 23 pairs are all copied. A copy of each pair is made and sent to the new cell. But when sex cells are created through a process called meiosis, those cells only receive one copy of each gene. So that pair is split into single copies of each chromosome. And then when fertilization happens, you get a pair from your mom and a pair from your dad, and that's how the chromosomes are mixed. And of course, we'll have dominant traits on some of the parental chromosomes and recessive traits on some of the parental chromosomes. And that's what leads to all of the different expressions of human characteristics that we see. So here on this slide, we have just a little bit of terminology related to genetics. Specifically, so if we look at a chromosome, the individual specific sections that control individual traits. So the section of a chromosome that DNA encodes for height or eye color or hair color, that's what we call a gene. The different versions of that gene. So if we say this is the hair gene, you can have different alleles of that gene, which is different versions of the same gene. So you might have a brown allele and a blonde allele and a red allele for the hair gene, the specific location of the hair color encoding part of that chromosome. And then the location, the physical location of that gene on the chromosome is what is referred to as the locus or the loci. And here we have the description of what we call genotype versus phenotype. So genotypes are what genes are actually inherited from the parents. So for the pea plant examples that Mendel did, if we call purple dominant and white recessive, then you can have three different genotype possibilities. You can have genes that are both the dominant purple genes. You can have one dominant purple gene and one white recessive gene, or you can have both of the genes be white recessive genes. Now, that is different from phenotype. Phenotype is what is actually expressed in the organism. So phenotype in this case is basically, what color is it? Is it the purple color or is it the white color? Well, for the first two genotypes, the ones that have either both dominant genes or one dominant and one recessive, because the dominant is present, that means that we have a purple phenotype there, meaning you have purple flowers because you have a dominant purple gene. Only when you have the two recessive genes do you see the recessive trait. So we don't see the white flower phenotype until we have the double recessive genotype. So now, thanks to the work of Mendel and those that followed him, we can now sometimes use tools like we see here, like Punnett squares, to predict what traits or what possibility is there of inheriting certain traits from a given set of parents. So the example here is for color blindness, and because of the way color blindness works, a man can either be colorblind or not. A woman can be a non-carrier or a carrier or colorblind, and in both cases of the carrier and non-carrier, the mother has normal vision. They can have the recessive trait and still have normal vision. Men, like I said, if they have any instance of it, they're colorblind. So, and you can see if we line the different recessive and dominant traits up, that allows us to see how many of our offspring of these different couples might have or might experience color blindness. And of course, this can be done for a lot of different positive and negative traits, disorders and traits that people want to see in their children sometimes can have the possibility or the probability of them determined by looking at something like a Punnett square. And then we've actually, this was just another neat graphic I thought, shows different chromosomes, the different human chromosomes, where different disorders are located, what specific chromosome we might see different genetic disorders or inherited disorders arise from. I just found that one interesting. With all of that said, human inheritance and complex inheritance, complex genetics, is a little bit more complex than the original models shown by the P plants. Of course, humans have much variation in the characteristics that they show. The earlier example that I said was eye color. Brown eyes and blue eyes and green eyes and hazel eyes. Hair color is another great example. We have blonde, brunette, red, black, and so on and so forth. And height is greatly varied in humans. We have people from max of 5 foot all the way to max of 7.5 feet. So there's a lot of variance in the different characteristics of human. It's not as binary as pea plants where you can be tall or short, you can have brown eyes or blue. The interaction of our genes is a little more complicated than just dominant and recessive. A lot of times you have some genes that are co-dominant, which means neither one really takes hold over the other. You can have some traits that have more than 2 genes. For example, blood type has 2 different alleles associated with it. So you can have more than just 2 combinations of blood type. And we see that with A, B, and O blood type, along with positive and negative. So there's a lot of different variations in humans. Their characteristics are often controlled by more than 1 gene, and we can't predict as easily because of that as we could with the pea plants. But the work of Mendel was still very, very important because it allowed us to begin the understanding that allows us to further evaluate kind of how our characteristics are passed down generation to generation. So now to finish out our discussion, we're just going to talk about some of the recent advances in genetics technology and where we're headed in the future with genetics technology. This is probably one of the fastest growing branches of science, and one that will really, really impact future therapies and medicines. So what we refer to as our genome is the entire code for each of our chromosomes. So chromosomes are built of DNA molecules, and DNA molecules are linked together in a double helix that are kind of, they stick together. You got a column here and a column here, and then stuff in the middle, and it sticks together like this. And each of these linkages is what we call a base pair. And there are about 3 billion of those linkages in your chromosomes, and they all have a different code for them. We code them with letters A, G, T, and C. And so we worked for a long time. It took, they started in about 1990 and they finished in 2003. So 13 years scientists worked to decode that entire genome, to be able to write out every one of those base pairs in a long code-like fashion. And we were successful in doing that. Something interesting from that is all of us have about the same 99.9% of base pairs, so about 2.99 billion of our base pairs are the same. 0.1% of those base pairs are different, and that is where we get all of the variation in human characteristics. That's where we get people of different heights, people of different eye colors, people of different skin colors, people of different all kinds, any characteristic you can think of. It just comes from that 0.1% of DNA. We are otherwise 99.9% the same. That's a good thought, isn't it? When we were able to do that, now that we're able to do that, we're able to further investigate regions of these genes and find out where errors are that lead to genetic disorders, that lead to higher chances of cancer, that lead to higher chances of heart disease, and maybe even one day we'll be able to go in and fix those things. And like we just said, one of the reasons that we care about knowing the human genome is hopefully we want to be able to give therapy to, or maybe even one day cure genetic and chromosomal disorders. And so we like to make the distinction. People with genetic disorders have a mutation in one specific gene. And a lot of times that can be things like Marfan syndrome, cystic fibrosis, sickle cell. They're all listed here. Also, it can also be things like just a gene that has the wrong couple of base pairs that lead to a much higher risk of breast cancer or a much higher risk of colon cancer. So all of those things can be considered genetic disorders. People with chromosomal disorders have extra copies or are missing a specific copy of a chromosome. Those are things like Downs we mentioned earlier. Turner's and Klinefelter syndromes are also examples of those. Those would be a little bit harder to correct, but maybe our technology will get there one day. But when those cells are divided and reproduced, that same chromosomal anomaly is reproduced along with it. So that is a much harder um, thing to correct. Whereas with the genetic disorders, if we can just fix one or two of those base pairs, that gets copied over. The fix gets copied over time and time again. All right, so that brings us to the discussion of current biotechnology, and that, of course, is going to include a little bit about genetically modified organisms, which is kind of a big, bad, scary word in the grand scheme of genetic science and overall in the news today. We'll talk about, just for a minute briefly, how advancements in biotechnology have made a lot of new things possible. PCR in particular, I'll talk about PCR for a second on the next slide, and some neat things that go on with polymerase chain reactions. We'll talk about gene insertion techniques to allow us to genetically modify organisms such as food crops and bacteria. And we'll say for a minute, not all GMOs are bad. We'll talk about that on the next couple of slides. Perhaps one of the biggest developments in science in the last 50 years has been the development of the polymerase chain reaction. So it's a kind of big sounding word for a process that just means we take a piece of DNA and we copy it over and over and over and over again. The technique has really been perfected to this at this stage, though. That's what's really nice about it. We are able to do this with very good efficiency these days, and it has allowed us to to really make a lot of advances in certain areas of science. For one, we can determine the genome of an unknown virus or bacteria very quickly. We're used to, before PCR, before we were able to replicate DNA really quickly, if we took a virus sample or a bacteria sample, we might have to let it grow in a culture for days or weeks before we could see it and determine what it is. Now, if we can just collect DNA from a sample, we can take that DNA from the bacteria or virus, perhaps from somebody's nose or infection or something, and we put it in a PCR machine and we replicate that DNA over and over and over and over again. And then we can test that DNA to see, is it part of the virus or is it part of the bacteria that we are looking for? That's how the most common or the most powerful test for the COVID virus during the pandemic worked, the PCR test. They would take a swab of your nose and they would replicate whatever virus was causing that those cold symptoms. They would replicate it over and over and over again, and then and then when we had a big sample of the DNA, we could compare it to the DNA of the known virus, and that would allow us to determine whether or not a person had COVID. Also, what's the word? Perhaps more interestingly, there we go. Lots of people these days love their crime dramas, right? So DNA fingerprinting is all thanks to the PCR reaction. So what happens with DNA fingerprinting is when you collect DNA at a crime scene, a lot of times it's just tiny little amount, and so the PCR reaction allows us to amplify that. We take that little bit of DNA that we found at a crime scene and we put it in a PCR machine and we make many copies of it. And after we make many copies of it, we run it through what we call gel electrophoresis. And so gel electrophoresis is this diagram looking right here. And everybody's DNA will come out looking just a little bit different in gel electrophoresis. And so we can treat it like a fingerprint. And we can see here what we'll do is we take the DNA we get at a crime scene and we amplify it a bunch of times and we run it through our gel electrophoresis. And we see what DNA we found at the crime scene and we compare it to our suspects. And we also compare it to our victim to make sure that we're not getting just our victim. And by that, we can figure out who was at a crime scene and who was not. So that's at least an interesting part of the science that we're seeing around us today. All right, so I said I would get here. I know how people feel about this word. I know how people feel about this topic. Because it stays in our media and it can be a big, bad, scary word. But not all GMOs are bad. So genetically modified organisms result from when we insert new DNA directly into the genome of some organism. So we put the DNA in some organism, and that DNA is incorporated into that organism's genome, and it allows us to do new things with that organism. In fact, I told you they're not all bad. Bacteria have had insulin-producing genes from humans inserted, and when they do, they start producing insulin. We can gather that insulin and give it to people who need it. That's where all our insulin comes from these days, in fact, is genetically modified organisms. Before that, we had to grind up the pancreases of much more cute and cuddly animals to get their insulin so that people could use it. So it's a lot better now that we can get it from just bacteria and not animals that we tend to like more. Perhaps more against the grain is the genetically modified organisms of our food crops. So of course we've been able to genetically modify, corn's a great example. Corn's the great American grain, and we have engineered it to death. We've engineered corn to be much more drought resistant, so it takes a lot less water to grow. Pesticide resistant. They've engineered corn that can have Roundup sprayed directly on it so that all the weeds die out around it, but the corn is unaffected. We've engineered them to have more grain and be of bigger size. And all of those things can be considered good for a food crop. It's better if we can produce more food for more people without having to worry about things like, is it going to rain, or is my field too weedy? But a lot of people worry that these genetically modified organisms may affect us somehow. It may I don't know, give us cancer or change our DNA. And that there's no real way for the DNA of corn to be affecting your DNA. We just use it for fuel. We break down the sugars and stuff and everything else. Well, you know. There has not been any, these have been studied for quite a while now, and there's not been any adverse indications from the consumption of genetically modified foodstuffs like corn and wheat and such. That's not to say they're not without their negatives. There are, when we have engineered this, some of these have cross-pollinated with different plants and imparted pesticide resistance or bigger size to plants that we don't want to have those things. And so it may still affect the environment around us. I'm not going to sit here and say that they're completely they're completely good and don't have any negative consequences. I don't know that, but I do. It has been shown for the most part that consuming them is just as safe as consuming any other food. And finally, I will end with this. So you may or may not have heard, but we are getting closer and closer to really perfecting new technologies called CRISPR and some others that are closely related. And with these new technologies, we are going to be able to go into people and snip out certain sections of DNA and put in certain sections of DNA that we want. It's not perfected yet, but it will be sooner or later. And when we do, I will say that one thing will happen. It will lead to cures for some genetic disorders. At some point, we're going to be able to snip out a bad section of DNA and insert a good one, and that's going to give somebody some relief from some kind of disease or inevitability. And that will be, I failed to see how that may or may not be a good thing. I think it will be a good thing. However, and I'll leave you with this thought, that does mean that at some point we may be able to genetically engineer traits into people that are already living or future children that we want. Now, whether that's an ethical thing or a good thing will certainly remain to be seen, but it is probably something that will be questioned of in our not too distant future. Uh, Thanks for watching the genetics presentation.